All right, well, we're ready after a wonderful lunch to dive into a discussion of ecological insect management. And it is my pleasure to introduce a relatively new colleague at UW-Madison, Emily Bick. Emily is an assistant professor of precision pest ecology for field and forage crops at UW-Madison. She's been at multiple universities, Cornell, UC Davis, and then more recently has spent some time in Europe. And the Bick Lab Research Group focuses on applied agroecology, digital entomology through modeling technology, and spatial population biology, with the goal of their research to develop tools and strategies to support data-driven precision management practices. So Emily is going to be talking today about ecological weed management and some of the exciting developments in digital entomology that she's doing in partnership with graduate students, and touching upon armyworm, which was a challenge for many of us. So please join me in welcoming Dr. Bick.
Hello, everyone, and thank you for the introduction. I know I probably get the slow crowd post-lunch. That's okay. I can bring the energy, and hopefully you all will meet me where I am. That was a fantastic introduction. So I am the brand new, a year and a half in, assistant professor of precision pest ecology for field and forage crops. So what does that mean? Well, I try to use some of these kind of modern methods, some digital methods, some modeling, some sensors, but modern methods like text. And we'll be talking about the insect pest text alert service that we're launching in Wisconsin later in this talk. And I try to communicate really important timing and spacing specific information about insects. And this is particularly important in organic or low-till or kind of ecological farming practices. My background is in entomology. Ooh, there we go. So this is kind of, I like to really start this story out with where the saga begins. And can everyone hear me all right? Is it too loud? We're good? Excellent. Okay, so where the saga begins. This is a map of all of the places I've conducted field work across the US, Mexico, quite a few places in Europe. I should probably put Indonesia on this list now that I've been out there doing a little bit of field work. But this is really how I came into being. I have 3 degrees in entomology, so I'm really an entomologist. I am from New York City, so I'm an entomologist lost in a cornfield these days. But you know, we're trying. I started in upstate New York. I was at Cornell for my undergrad. I was really mosquito focused. And this part is critical, right? Because mosquitoes are like the entry insect for entomologists, right? Everyone can relate to mosquitoes. We can relate to the diseases that they transmit. We understand they're scary, they're bad, sometimes most cases, right? And we can start thinking about how they move and how they impact people. And that's really my relationship with insects. I like them, I think they're very nifty, but I really care about insects directly impacting people and the way that we work, the way that we farm. After Cornell, I moved to the Midwest. I was actually Minneapolis located for about 2.5 years. I was working on emerald ash borer primarily, which is an invasive tree pest. Oh my goodness, it's a major, major problem across the US these days. And I was on the forefront. In fact, I am first author on the largest insecticide study for emerald ash borer, showing that you could actually treat a tree and have it outlast that infestation coming through Chicago. So that was really interesting, especially because during that time, there was this group out of Michigan State University that was doing something called the SLAM model that they developed, a slow emerald ash borer mortality model. And the thought was kind of like herd immunity in people, kind of like something I was thinking about when I was thinking about medical entomology, was if you could actually slow the mortality of the trees down a little bit. In this case, they were treating 10% of the trees with a emectin benzoate, a insecticide. If you slow it down just 10% every year, randomly across an urban forest, you could actually have the tree outgrow the insect. They use this really cool simulation model. Think a little Sims game where they would put in these really accurate scenarios. Does the insect win? Does the tree win? Do they kind of tie? And they came up with this kind of optimization of how do we actually manage these trees in the Midwest? And I thought that this ecological modeling thing was such a nifty concept. They almost reached this herd immunity level where if you were treating 10% of these trees, of ash trees across the ecosystem, you could kind of reach that herd immunity where the treated trees protected the untreated trees. So that was a medical concept that we were bringing over. I thought that was so nifty that I decided to do my master's and then my PhD on that concept, on using this data about timing and spacing, these different types of interventions, about knowing biology of an insect, biology of a crop, or biology of, in my next case of working an invasive aquatic weed and its biocontrol, and trying to optimize the way those pieces interacted in the ecosystem. And this is where I kind of got my start for kind of the automation game, for trying to provide farmers and decision makers and stakeholders with better data quality, more timely data access. So after I did my master's on these invasive aquatic weeds and an insect biocontrol agent, this was water hyacinth, Neocotina brucei, I then went on and started working in organic strawberry systems. And I was working on a trap crop for Lygus hesperus. So how do you use alfalfa as a way to pull these Lygus bugs out of the crop? And I was asking questions like, where do you put it? We had this crop that was in the ground for about 9 months out of the year, or actually producing for about 8 months out of the year in California. So we're managing these both alfalfa stands, we're managing the strawberries in this case, we're trying to optimize this placement of alfalfa crops. And what I found out was in this particular case, alfalfa wasn't acting like it was supposed to. Instead, it was providing a source of beneficial insects, and those beneficial insects were then spilling over into the landscape. So I kind of said, oh, wow, now is the time where we need really high quality insect data, where me going out, driving a couple hours, taking some samples, driving a couple more hours, taking more samples, wasn't quite doing it in terms of providing data for farmers to make better decisions. So that's when I moved off to Europe. I got a Danish Innovation Fellowship, and I was working with entomological sensors. And I swear we're going to bring it back to things that are relevant to you in about a minute here. But in Europe, working on these entomological sensors, you basically had a laser that was shining out a beam, an insect would fly through, and you would get this alert that the insect is there. You could use some nifty kind of cutting edge machine learning models to figure out maybe what is it with some amount of certainty, some amount of uncertainty. But it provided this incredible kind of low labor automation piece. So the back end of my talk is trying to develop that type of sensor, a type of insect sensor that provides really, really cheap access to insect phylogeny. So how do we find out that insects are in your field? Okay, then I was invited back to Wisconsin a year and a half ago, and now I'm focusing on field and forages. Oh, there we go. So at the crux of it, my lab is trying to develop this fundamental understanding of insect population dynamics. It's basically how insects are moving and changing over space and time. We're trying to translate that understanding into these novel tools and strategies that enable data-driven, and you can fill in the blank here, sustainable ag, organic ag, precision ag, call it whatever you like. More data equals more power, equals better decisions, equals more successful agriculture. And the approach we like to take, we term digital entomology. And this is my brand new lab logo that got finished yesterday. So tell me on the feedback forms whether you like it or not. We're the original digital entomology lab. We're trying it out. But the digital entomology is really confusing because it's brand new, right? We don't really know what it means. But from what we're doing once again, as the field and forage crop entomology specialist in extension at UW-Madison, what we're trying to do is combine sensors, combine those simulation models, and use some fancy new tools, machine learning, maybe sometimes classical statistics, to then figure out what insects are doing and help farmers make better decisions. And the really critical piece is we view all of the outputs of these sensors, of these models, of these algorithms through that lens of how insects are moving and changing over space and time. And I'm gonna keep hammering in the space and time bit. Now, when it comes to insects, I want you to think a little bit about this. What do you think matters more, timing or spacing? And now we're gonna pull in the participation. Raise your hand if you think it's timing. Timing matters more. Okay. How about spacing? Raise your hand if you think spacing matters more. There's some spacing people in the audience, but the vast majority of folks raise their hands about timing. Now, timing is, I would argue, way more important than spacing. And even though I really like the where do you put it, how do you manage it, those types of questions, timing is the critical piece. Because if you miss the timing, you're also missing the spacing. So spacing is dependent on timing. And okay, we're backing it up, and we're talking about some insect facts here. So that was kind of the overview of what my lab is up to. Now we're gonna talk about insects in particular, and then we're gonna talk about insect pests. I'm gonna walk through some true armyworm kind of frameworks, and then walk into an insect pest text alert service that we're launching this summer. I'll have a link in case folks want to click that link and sign up. And then we're going to be walking through some of these newfangled sensor gadgets that eavesdrop on insects. So less than 1% of all insect species are considered pests. So the vast majority of insects in your ecosystem, in your field, outside of your house, are really not necessarily important to people, right? There's some beneficials, we'll talk about them in a minute, but most of them, in fact, look at this diagram over here, most of these insects are just also occupying the world that we live in. Morphologically, when we talk about insects, we talk about these little organisms that have the chitinous exoskeletons, the hard outside. They've got 3 pairs of legs, 2 pairs of wings when they're adults, mostly, 3 body segments, a head, a thorax, and abdomen. They're symmetrical, right? So you can tell them apart. But they can take this massive difference in size and shape and function in the ecosystem. But from an ecosystem perspective, there are these amazing benefits from having insects. Pollination, right? Like that bee up top, right? Bees are very specific pollinators, but so are moths and butterflies, so are beetles, so are some true bugs, where they help assist bringing pollen to where the plant can actually reproduce. They're at that intersection there. There's this natural pest control. So if you do nothing in an ecosystem, you will have a significant number of predatory insects, such as ladybugs, or parasitoid wasps, things like spiders or other predatory beetles that can help control your pest. And that's with doing nothing. In fact, adding pesticide tends to hurt those populations more than doing absolutely nothing. You have decomposer insects. So these are insects that break down organic matter. They help with the nutrient cycling. They enhance soil fertility, making more productive crops. And the more diverse your planting area is, the more diverse your insects will be in response to that crop or that area. There are insects that help with soil aeration. So burrowing insects kind of create this channel, which allow for water infiltration and better nutrient absorption. And there's kind of the biodiversity support, right? So agriculture is, at least if you look at the land use, I think 34% of the land in the US, right? So we need biodiversity of our insects in agriculture to kind of maintain that ecological resilience against pests, where the more occupied, the more numbers of insects in this niche space or in your agricultural field, the less likely a single insect is going to come through and dominate like a pest species, be a challenge. And insects are widely successful. In this case, diversity really does mean success for them. There are over 1.5 million named insect species, and estimated up to 4 times more than that we haven't even looked at or an entomologist hasn't formally named. I'm personally very grateful that I'm not one of the entomologists that has to look at insects and name them all the time. That seems like a lot of microscope work and not enough field work. They have this wide variety of food sources, right? They eat pretty much everything because, we've seen insects eating styrofoam recently, right? And they do that because of the diversity of types of insects. Their habitat is most places. That's really where they live. They often don't live in salt water, but that's pretty much it. We find these ice striders in Antarctica, and they're really successful because they have the great ability to move, right? The adults can fly. The immatures, hey, sometimes they move around a little bit, and almost never as much as the adults can move around. And then there's one last bit, and some insects can actually specialize on 2 types of food sources where they're immatures. In this case, insects that have larvae that go through that complete metamorphosis. Think not true bugs, but beetles or moths and butterflies. Their immatures specialize on a completely different food type than the adults specialize. So we're going to talk about true armyworm, and the adults are nectar feeding. They're not damaging for the pests. Where the damage comes in is when they lay eggs and the eggs turn into larvae, and the larvae are just incredibly destructive. And if you take a look at all of the insect pests across, even though only less than 1% of all insect species are pests, if you take a look at all of the insect pests across all of the food-growing regions across the entire world, on average, about 20% of all crops are lost due to insects. That's pretty significant. That's after all of the chemical inputs that we put into this type of system. So they're successful because of their great diversity, even though most of them are not harmful. And then we've got some good news and some bad news when it comes to insect behavior. And this is where I start thinking a little bit, and I hope you all will join me in thinking a little bit more about that spacing and timing set of elements. The bad news is across the globe, across space, across every single level, across a landscape, a field, a patch, insects are really patchy, right? They form these clusters. In this case, this is pollen beetles entering a canola field in Denmark where I was doing my postdoc, where at one stage they enter the sides, and then another time point they're just in the middle. So the red is where they're highly aggregated or grouped or clustered, and the blue is where they're, really not found that often. But then that changes, that almost swaps as you move on in seasonal time. So the bad news is they're really, really clustered or patchy or aggregated. And those patches shift, right? Which means that one year to the next, we're not going to have the same insect problems. In fact, that's a major, major issue, that they're shifting around, and it's really hard to figure out where they are and how to find them and how to do something about it. However, the good news is insects cluster, right? So we're flipping this on its head a little bit. Insects are actually clustering at different spatial scales, and they do so at a fairly consistent, predictable rate, right? So if you take a look at, we were looking at potato leafhopper and alfalfa this summer with some collaborators, and it turns out the adults tend to come into the edge. They lay eggs on the edge of the field, and they move into the center, and then you find them all throughout the center. But the larvae are really aggregated at the edge, which means if we know something about their pattern, about the spatial placement of where these insects end up being, and then we can adjust our time frame with maybe real, live, updatable information, possibly automated, oftentimes probably not automated, then we can start adjusting our management practices, where if you're trying to target potato leafhopper and alfalfa, you want to hit the adults and you want to hit them at the edge while they're immigrating into the field and starting before they fully colonize. So growers can actually take advantage of that where and when insects are and take a look at their behavior and adjust The placement and the timing, really the timing, the placement, as we discussed, timing is way more important than spacing, to try to mitigate their damage. Okay, so that's kind of the good news and the bad news. And you'll see when I talk about my pest approach, this is very much a strategy that I use with students, a strategy that I use with adults, a strategy that I use with everyone. I try to take the strategy of teaching a man to fish rather than giving him a fish. So this is the approach that we have when we're trying to figure out what do we do about an insect pest. And I know this is simplistic, but stick with me here. We like thinking about, or I like thinking about who the pest is, what's it doing, when's it arriving, where's it going, and why is it in our field? Why is it in the field that we care about? And I'm gonna walk through this idea, this concept with true armyworm. This way, when a new pest comes in, let's call it soybean gall midge or soybean aphid, we can start asking the exact same questions and addressing them appropriately. And this all ties really nicely back to management takeaways. Or if we know these pieces, then we can much better precisely manage our insects, which means we'll be much more successful in the management, especially when we don't have access to conventional pesticides. So this is a true armyworm larva. It's on a corn leaf right now. It has a light brown head. It's got some stripes on its back. And I think Roger, who took this picture, is in the audience, so your name's up there, Roger. But the who. So true armyworm are migratory. That's really important. That means that the adults in the first generation come in on storm fronts. It also means early on that we, or I, or someone in DATCAP, Department of Ag, Trade, Consumer Protections, can actually talk to folks down south where they're migrating from and ask, is it a bad year? Is it a good year? And we can have that early warning heads up. Okay, migratory, that's really important. They're sand colored, the adults, an 1.5 inch wingspan. They've got these white dots on the front of the forewings, and the back wings are dark. However, a lot of moths look exactly like this. So it's not really the adults that we have to be super careful for identifying. Or you can trap for them. You can put out a pheromone trap and catch mostly these adults. Okay, critical bits about the who. There are 2 to 3 generations per season, mostly 2 in the state of Wisconsin. And it's really the second generation that's the much larger problem. We'll talk about when they're a problem in a little bit. But that means if you get them in the first generation, they're flying in on these long-term migration flights. The second generation hatch and they pretty much stay where they are in the field. So if you manage them early, you're gonna have much less of an issue down the line. So if you get their first generation, you're probably gonna stop or really, really suppress their second or third generation. These adults last for 5 or 6 weeks where they're mating, they're laying eggs, and the adults are just feeding on plant nectar. They're really not an issue whatsoever. Now, are the adults the issue? I'm good with long silences. So I see a little bit of head nodding. Does anyone want to shout out? No, excellent. No, the adults aren't really an issue. Well, asterisk that, right? So the adults are an issue because they lay the eggs, and the adults are a bit of an issue because they get to choose where they're laying eggs. They get to choose where they're ovipositing, right? Adults really like laying eggs on grassy areas. So that means if your field has a lot of grassy weeds in it, it's going to have a much worse true armyworm population from that set. Oh, yes, so the adults get to choose where they're laying the eggs. The larvae, while they're mobile a little bit, they're not mobile in the same way the adults are mobile. But because the adults get to choose where they're ovipositing or laying their eggs, and because they really like grassy environments, that means if you have a field that has a lot of weeds, that has a lot of grass, and there's a big influx of true armyworms, you're going to have a much larger issue down the line. What's the damage? Why do we care about this? Well, the larvae do primarily feed on grasses, but infrequently, and yet still significantly, they feed on cereals such as small grains and rice, forage grasses and turf grasses. This is what they do. right? So if you're seeing all this chomping over here, or if you're seeing, especially during the second generation, kind of that nibbling on the edge of your field, on the edge of the leaves, that leaf defoliation or kind of head clipping, that's really important that you go and you look for caterpillars, right? Because timing's really important with these guys. And I would suggest checking all fields closely, looking at both leaf defoliation and that head clipping behavior. Now, armyworms, who here has had problems with armyworms? Lots of last season, everyone nods, head nods. Yeah, last season it was a tough year for armyworms. Ooh, yes, so no, and armyworms, especially if you're in conventional fields, are really only susceptible during the first 2 or 3 instars. So if we miss the instar timing, if you know that it's there and you miss the caterpillar timing, they're not going to be particularly responsive. And I know there's at least one organic BT product out there that you can use on corn. I'm not sure if it's labeled on other grains, but timing is critical for these critters. Timing, there we go. Now we're cycling through. And I think I had a graphic, but we lost the animation here. This is a little animation of what I was doing in Denmark, right? You shine a light out, an insect flies through, and you can maybe get better timing. But generally speaking, in the upper Midwest, this is what happens. Sometime in March, those insects come in, they lay eggs, the eggs start developing April, May, so you have some of that early feeding. And the plants look more like the first picture of that head clipping than like the second picture of the leaf notching. They pupate and they turn into adults between June and July. That timing is extremely dependent on when that first influx, when that generation came into the field. Those moths, June, July will lay eggs, and mid-July, late July, possibly a little bit further out, that's when true armyworms are a larger problem in the state of Wisconsin anyway, the upper Midwest. They pupate, they can overwinter, but most of them die off and then they come back in the next year. And why is this timing super important? Well, it helps inform when we take actions. And okay, where? Here we go. Movement rules the day. So after we've got the timing in, we can take a look at some of the adult behaviors and start to predict, why your field, why is it damaging your field? So these adults, they come in and they lay eggs primarily on grasses. They can also migrate from pastures over. So if there are a lot of them, they can exhibit a spillover behavior where they're moving from one area to another. So they can migrate from pastures, spring-seeded grains or small or grassy hayfields. They can invade the outside rows of corn as well. And knowing something, and this is when you're approaching a new pest, not necessarily true armyworm, knowing about what the movement of the insects are doing will indicate where you actually need to go scout, where you need to actually go take that action. And then why? And this is what I think everyone across the state in Wisconsin had last year. So surprise, we have armyworms. So why are they in your field? Well, the first generation damage is often really difficult to detect. So scouts often miss it, right? It's if you see chomping but you don't see an individual, it could be caused by a number of things, number of organisms. So it's also a lot more scarce than that second generation, where the number of eggs laid by the second generation, the number of larvae are much more prevalent. The larvae also prefer grass. So there's this really funny hypothesis called mother knows best. So what the larva eggs are laid on, the almost across the board, insects will prefer what their eggs are laid on, what they taste first, because they trust what mom did. They trust that mother knows best, that the food source they were laid on is their actual preferred food source, regardless of nutrition content and all sorts of other things. So because larvae prefer grass, corn planted after cover crops may have higher populations. So that means if you have a field with corn planted after a cover crop, it just means to look more into that field. And then also, unfortunately, more weedy fields have higher numbers because of the grasses that pull those insects in. or those grasses serve as like a shining, flashing neon light, right? Come here, come lay eggs. And then when the larvae run out of food, they move on to the crop that we actually care about. Okay, so now what? Let's kind of pull back a little bit and understand how to use that who, what, where, when, and why to understand something about ecological management. So true armyworms are the who. Scout is the what and where. So scout your corn and wheat fields, especially grassy weed with grassy weed growth or corn that isn't tilled. It's no-till into alfalfa and fields with grass cover crops. You could also figure out when with the Gride models, though those do not account for when insects come in that first generation on storm fronts. It's something my lab is working on, but we're gonna be a couple years out before we can roll out something that integrates both the Gride models and scouting trapping data from DATCap. They've got this amazing field notes section. And then we're putting out this year with DATCap and with UW Extension, Josh Camps is extremely helpful in this, a insect pest text alert service that tries to take the information from DATCap scouting and sends it as a text specifically in your region. Hey, guess what? Go scout. But ecological management-wise, there's a fair amount of biocontrol. I think there's a tachinid fly that you can actually purchase and release in your field. Those help keep armyworm numbers down, as do birds, as do bat boxes, as do toads and skunks and some domestic fowl. In fact, in Indonesia, I saw folks literally put out bat boxes. They did this a little bit in California as well, around the field to actually drop and severely suppress the numbers, because if you provide habitat and you're providing food, the bats will be there and be very happy at eating your pests. Culturally, we try to keep grassy weeds controlled. We try to avoid low wet areas in rotations following sod, or you can plow if you need to. Herbicide and tilling sometimes drives up armyworm numbers, sometimes drives armyworms from one crop into more susceptible crops. So beware that you can have spillover if someone does something adjacent to your field, especially in the moth stage, more so than the larval stage. Now, I mentioned this a little bit, but I'm really, really excited to launch this insect pest text alert service. My name's on here, but that's only because I was too lazy to go get a DATCap logo and an extension logo. But basically, we are following in the steps of folks much smarter than me out of Iowa State University. Aaron Hodgson's group launched this Iowa Pest Alert Network, and I learned about it when working with Aaron this past year, and I think it's such a good idea that we're just following suit. Basically, DATCap goes around and puts 450 traps. In fact, we might have volunteers in the crowd that manage these traps. They have pheromone traps, they have black light traps, and then they have people running around the state that are actively scouting for insects. And this is the list of 10 insect species that we're gonna be targeting this upcoming year. We're also, my lab, we do a lot of kind of mathy spatial things where we're taking a look at where all the traps are right now, which is this map over here. We're seeing where there's missing in the missingness and where there's over trapping across the state. So if there's a high amount of, let's say, the crop that the insect goes to and a low amount of traps, so if there's trap autocorrelation, aka those traps are reporting the same numbers every year, you can pretty much delete those traps. And we come up with a kind of a meaner trapping network that has better coverage in the places that we need it and less coverage in the places that we don't. So we're updating the trapping network this year in response to real field needs. And then the actual service, there's going to be a sign-up page, which I'll have a QR code up for in a moment, that basically asks your name, your phone number, your email, and which regions you care about. A DATCap network of traps, that's the 450 traps and walkarounds that they do, monitoring those 10 pests, where basically DATCap, Josh Camps and I will get on the phone every week and say, okay, what is significant to tell folks about? And then we'll send a text out that says go scout or heads up. It will never say go spray. It will never say go do something. It's basically go look in your field. We think it could be an issue right now. And then it'll link to an extension web page, which will have someone either in my lab or an extension that can answer questions about it, their phone number, a set of extension resources that we've briefed beforehand, as well as the link to the field notes page, which DATCAP runs that has those actual trapping numbers. I think we're also building out some dashboards, so it'll be a little bit easier to access that DATCAP information. We think this will majorly fill in a missing gap, so people will be a little bit less surprised. And this is all because timing matters so much when it comes to insects. This is the sign-up link. Sorry, I can get back to this at the end. There's a QR code. You can also easily find it on my web page. I'll let folks take pictures here. Right now it's a Google form as we're negotiating which texting service we're using right now. Cool. I'll wait till all the phones go down. Awesome. So that is about as low a tech a solution as we can do, as folks in my lab can help facilitate the spread of information that we are not collecting, that DATCAP is collecting already. However, what we also focus on is that automation of monitoring, right? So insect sensors have been around for a really long time. We have these optical sensors, which are camera-based often, or they could be LiDAR, where you shine a laser out and an insect flies through and you collect that information coming back. Entomological LiDAR has been around since 38, so it's actually quite an old technology. It just recently got in the hands of entomologists like myself, and we kind of went crazy with it. We've got these smart traps or open air cameras, so cameras either aiming at a sticky card, which you have to go and change anyway. FMC has a smart trap network with like 4,000 of these traps that we're working with, or open air cameras that try to identify insect in flight. There are also a number of audio sensors that are just these air microphones that we've been calling them. So one of these put in let's say a cornfield. They usually can get things like wing beat frequency and something about the time of day that the insect's going past. But segregating out by wing beat frequency is really hard when you're not talking about moths and you're not talking about mosquitoes. Most things have a wing beat frequency around 200 hertz. So it's a little bit hard to untangle that bit. But this was the sensor that I was working on. And it worked really nicely because it shone that light out. The insect flew through the beam. And when the wings were out mid-flap, we got more light reflected back than when the wings were down. So from there, you could get this graph over here. Let's see. Hey, that works. So then you could get you could actually get the wing beat frequency, which is the distance between those spikes. You could get a body size to wing size ratio that they usually just measured smack dab in the middle. And you could get pretty close to species, or you could just train a machine learning algorithm on it and get to species. And we showed not only was the sensor great, but it was significantly more efficient, right? So this sensor caught 2 to 3 orders of magnitude more insects than I could do when I was going out and beating 450 plants every single day, tracking pollen beetle immigration into a system. It beat water traps by about 3 days, assuming you're checking your water trap every day, which I know most people are not, and I wish I didn't have to either. But hey, that's the researcher job. And it beating plants for pollen beetles by about 5 days. So we know these sensors are a really good idea. Oh, and we just have an eLife paper out saying that you could actually ask how different the insects are and get a biodiversity richness number or metric. It says how many species are in the field. And it was more correlated. It was a very, very good metric there. But I knew when I came to UW-Madison that we really needed to do these kind of cost-effective sensors. So instead of paying 10 grand to rent one sensor for a season, we know one sensor is probably not representative of your field or your system. We were really targeting something like $16, $15 sensors. And you might have seen this news that keeps getting amplified. I think my grad student is winning the marketing game right now. But I invented something called the insect eavesdropper, which is a little clip-on guitar mic. It's a contact microphone or a piezo microphone. It clips to a substrate or sticks to a substrate, and it vibrates when that substrate vibrates. You plug it into some things, and you put it on a mini computer that basically presses start, stop, and save, and then you deal with the data offline on the device, which looks like that right now. But basically, we can hear as insects are actively feeding, but not walking on the plant. You could also walk it around the field, and we've termed that the rambling eavesdropper if you're trying to do subsamples. So we had to test it out, right? This is a brand new technology, never before been done, though we're kind of standing on the shoulders of giants because people in the 80s put gramophone needles and seismograph needles on plants and they could hear these types of things, and $2,000 laser vibrometers were aimed at plants. This is just a really cheap way of doing things that folks have been doing in the lab for a bit. But we tested it with these large-bodied insects, right? Tobacco hornworm. Who's seen a tomato hornworm or tobacco hornworm? These things are giant. And then Colorado potato beetles, right? Those are also nice, chunky individuals there. And we also tested it on what we would target with this type of sensor. So the small-bodied insects, European corn borer. These are second instar, 11 millimeters. And they're boring through a whole bunch of different parts of the plant. And northern corn rootworm. So these are larvae that are actively feeding on the root zone of the plant. And well, we learned a lot about insects moving and not necessarily staying where you put them, right? The tobacco hornworm kept escaping and really liked hanging onto the wire, which was not what we wanted it to do. And you had to put the Colorado potato beetle and the control plant in a cage, or else they'd freely move between the control plant and the treated plant. And we couldn't just use a single corn rootworm larva because it would likely die. So we had to actually inject about 2,000 eggs to get there. But in every single case, we could detect insects directly feeding on the plant, essentially using the microphone as if the musical instrument instead of a guitar was a corn plant. Yeah.
That was in a controlled environment, right?
Yes.
The sound of tractors, the highway, the winds.
You were so priming me for the next set of slides. So we'll talk about the other sounds as well in a little bit. And for each of these plants, we ran two, one without an insect and one with an insect. That's what this graphic is over here. Then we also asked the question, statistically, how long do we actually need a microphone clipped onto a plant? And the answer was 40 seconds. To be 95% confident that a northern corn rootworm was on a plant, if it was, we only needed to listen for 40 seconds. For European corn borers, it was 90% confident. When you drop down to 20 seconds, it's 80 and 90, like 92, 93% confident, respectively. So that's how we got to the rambling eavesdropper. And even though this idea was not invented, it was invented a little bit ago, but it was built slightly under a year ago. So we're still new and still figuring things out. But one of our really key questions is, can we get to how many insects? Not just, is it there, is it not there, or what species is it, which seems like that's working as well. But we're trying to relate the number of bite sounds to insect density. And even in that 40-second period, we're getting some variance, some differing amounts of those bite sounds. Okay, so then we get to the really critical question, can we identify insects? And after a lot of pre-processing, I've got 3 computer scientists in my lab, 2 grad, 1 undergrad. We were able to get to these species identification algorithms, where about 97% of the time, at least on the lab data, it got to the correct species. So that was pretty nifty, especially because it takes a couple seconds to run that algorithm. Okay, who here has washed corn roots before? I spent a summer doing quite a bit. Okay, not as many as I would like in this audience, because this was a real pain. So to figure out if corn rootworm larvae are an issue, you pretty much have to either dig the plant up mid-season and do a Berlese trap, so try to get the larva to escape down, or also at the mid-season point, dig the roots up and do a root damage rating. So my lab was already doing a number of these pesticide trials for companies, and we had to do this root rating. So we, on top of this study, actually clipped on a whole range of microphones to make sure that on the positive control, where there was an industry standard insecticide, and on the negative control, where there was nothing going on, we were still seeing that variance outside. And this is we're missing some animations here, but basically across the board, we could hear insects outside, and we could see a difference in the rate of feeding when we do this outside. We were only measuring for 80 seconds twice a day during when the insects were feeding. Other noises, unless they came through the plant, they weren't a huge issue. So rain was obviously a problem because you can hear rain through a plant, but tractor sounds, unless the tractor's crunching over the plant, which did happen at least once in our field trials, that ended up not being too much of an issue. Okay, the next critical question that folks always ask is, what about piercing, sucking insects? We're talking about insects that do a lot of these large chomps. And we actually went out and found these plants that were completely clean of soybean aphids, that were next to plants that were just completely, you can't really see it over here, but this is orange, like completely covered with soybean aphids. And I think Roger's been telling me I need to play a sound, but you can go online and find the sound. But in our field tests, we were distinctly able to show if a plant had aphids and if it didn't. And it kind of sounds like someone stuck a straw through a milkshake and is slurping the very end of that milkshake. So it kind of sounds exactly what an aphid, you'd expect an aphid to sound like. Which was really nifty. We also partnered with a whole range of other scientists that ended up helping us do some of this validation work. So Brian McCormick, whose entomology department head at Kansas State University, did a whole density study on Lygus hesperus, or it might have been Lineolaris, I need to double check, as well as soybean aphids and soybean borers. But we're working on the data processing bit. We're trying to get a pipeline that's robust and repeatable. We're formalizing some of those insect identification algorithms to see if we can test if our ID algorithms that we trained and really clean signals inside, if they worked outside. We're expanding the number of pests in Kansas and Iowa and Washington, Alabama, in the United Kingdom and in Norway. And my lab is really keen on figuring out that density estimate, that how many question, to see if we can maybe get to insect thresholds. And in the future, there might be a phone application. We're not really sure what it looks like. I'm an academic, but I very luckily have this absolutely brilliant MS student, Dev Maratra, who's a computer scientist who will probably take this idea into a company form and let me, play in the field with my insects. But we also have Vidit Agarwal, who's an undergrad, a bachelor's, doing data science and computer science at UW-Madison. Lawrence Still was head of data at the sensor company that I talked about, that I worked with when I was living in Denmark. And he's now on board once a week. He was actually visiting last week from the UK, and a postdoc who focuses on machine learning algorithms as well. So it's not just me pushing this thing forward. I have a team of very, very smart folks backing us up. And these are at least our close academic collaborators as well. And then I'm going to go through this really quickly. These are kind of long-term goals of the lab. I have a PhD student, Helen Oker, who's trying to figure out where do we put interventions. Now we know timing's more important, but something like where should we put an in-furrow insecticide, or where should we plant something with a trait, or where should we release biological control organisms. So she's making these little simulation games, right, that match real spatial data, potentially with sensor data if we have it. I think she's starting with corn rootworms, but I'm sure she'll move around a couple different species. And she's helping us create heat maps like this, where eventually there'll be a web application. You outline where your field is, and it'll tell you if you need to take certain steps for certain insects, and you want to be spatially explicit about those steps. Where do we take those steps? Where do we put those interventions? I talked a little bit about integrating degree day models and actual pest data, right? So we've got these great degree day models, but the pest may or may not show up. And we've got this great pest data, but it's a little bit inaccessible. But if you combine those two pieces, then we should have something like a pest weather map or an updatable pest map where we're using land use data and these degree day models, these known pest models. We're able to update those with known pest information so that your degree day model forecasts become much more accurate. We're also quantifying biodiversity. So we have a General Mills sponsored master's student who's kind of pushing this, how do we measure biodiversity or even functional biodiversity? How do you measure bees in a field? How do you measure parasitoids or predators in a field? And can we do it with really cheap sensors? We're using cameras and sticky cards and microphones right now for that one. But this is our team, and I think that's pretty much all I have. So thank you all for listening. And are there any questions?
All right, questions for Emily?
So how do we control our armyworms that have come in for several years, or coming into the roll crimp, or either coming into the rye first and then from there they're moving into the corn?
How do we control armyworms? And I'm going to repeat the question for online purposes. How do we control armyworms that are coming into rye and then potentially, when you do something about it, moving into your adjacent crops? That's the right question? So there are a couple things we can do right now. We can try to make that rye look super unattractive, right? We can try to make it look not like a grass. That's probably not going to work so well. We can support a lot of biological control. So that's the bat boxes or parasitoid releases, or in Europe, in Asia, they do a lot of something called banker crops, where you provide resources for parasitoid wasps, like a flower strip around your field. It also looks kind of pretty, but that actually allows those parasitoids to be supported when the armyworm isn't there and then move back in. And this way, you get a little bit of a time delay with that kind of response. But if you get it going early enough, you could hit that first generation and really suppress the second generation. I don't think the BT spray is labeled for rye, but I need to double check that. Are these all answers you've heard before? Yeah. One thing we were talking about at lunch was potentially seeing if we could do mating disruption, where there's a pheromone that exists for true armyworm, and we could potentially, moths use pheromones to find each other. So if you flood a field with a pheromone, and these things are becoming a little bit cheaper because you can replicate them in yeast now and not just, chemically. If you're able to flood a field with a pheromone, you could do mating disruption, like they do with some other moth pests around the world. But that's something we need to test before we can advertise it. Yeah.
Have you tried the audio sensor on storage?
Ooh, have we tried the audio sensor on storage pests? No, we have not. So there is actually a long and storied history on using microphones, and these are air mics rather than contact microphones for stored grain pests. And there was a lot of really successful work done in the 90s, but I do think that the algorithms have gotten a lot better since doing that work. So it's something that I was in a group that Erin was organizing on Thursday, talking about ways to support, let's call it food grade grains, and that seemed like a really good research direction.
Nice and quiet in there.
It is nice and quiet in there. That's true. Less tractor noise. Yeah.
As far as armyworms go, do they lay the eggs in the soil or above the soil?
They lay the eggs in the soil. I thought that it was on the plant when I.
Okay, they lay the eggs on the plant.
I think so. I need to double check though. Yeah.
So you have the eggs on the plant, and then they hatch into larvae, and the larvae be on the plant as well.
Correct.
Are the larvae mobile?
Are the larvae mobile? Yes, but not anywhere close to as much as the adults.
So they don't move nearly as much as they do.
Correct.
So they wouldn't move more than a few inches possibly? Or?
They will eat a crop down and move on to the next one, right? So they're called armyworms because of this kind of marching behavior that they exhibit. So they can keep going, but it's not, I mean, they might move from one field to another, but they're not going to go 3 fields in one generation.
Does heat kill eggs or larvae?
Heat would absolutely kill eggs or larvae, but the question is, does it kill the plant first? Yeah. Yeah. There also are egg parasitoids, but I couldn't find a commercial link for an egg parasitoid. Any other questions?
So is there a way to monitor the armyworms with the trap, so when, like in my farm, I'm up in Minnesota, so they came in into the winter rye, but the winter rye that we chopped off for forage, there was none there unless it was lodged. Where the hay cutter cut it off, it was lodged. Them are the areas we had the armyworms, and they moved from there. They ate all the leaves off of the winter rye. The head stayed there. They moved next door to the corn and decimated the corn. So I mean, is there a way to trap them to know then I should come out with the BT spray or what the neem spray or whatever, and I can get ahead of the problem before they do damage?
Yes, so is there a way to trap them? So one, and this comes from a different system, not this particular system, but certainly in rice for lygus bugs, you actually have, when you harvest, let's say alfalfa, and then you've got rice there, you actually keep a strip of alfalfa, and the lygus will then move into the alfalfa, and you treat that alfalfa before the lygus goes into the rice. So if you plant what I'm going to call a trap crop, which is a slightly more attractive crop due to either mechanical harvesting or due to a chemical spray, if you plant a trap crop, a good strategy could be to spray that trap crop, because they'll hit that first, and if you're monitoring it, then you'll catch them before they go into the crop that you care about, in this case, corn. It seems like a good strategy. I have not seen a single piece of literature on it for armyworms. That doesn't mean that no one's tried it, though. But if you email me, I can do some digging.
That'd be an interesting strategy.
Yeah.
So what would be a trap crop for that? I mean, your rye at that time is probably this tall. You know, my wheat is not near as tall. The next wherever on the farm also, and then the corn is shorter.
A different stage of corn, or at least some kind of sacrificial corn, would be a good trap crop option.
What they do for vegetable crops, and I hadn't thought of that as an option but then you can target and it'd be much more cost effective your BT or
Yeah. It's almost like interception.
Yeah.
Yeah. There. If you're willing to try, I can help. I saw a question back there.
Are there endemic species or native species that we can encourage to become part of our system so that we have a reduced risk of them ever building up?
Yes, are there endemic species or native species that we can encourage? Absolutely, and I'm going to go back to this slide over here. So there are a handful, in fact, there are quite a few. There are some parasitoids. One of them is a tachinid, here we go. One of them is a tachinid fly. I think it's like some red-tailed tachinid fly, which you can encourage. The way you encourage it is you supply it other food or resources when it's not looking for the armyworm. There's a Burconid wasp that's an egg parasitoid. It's the exact same thing, and that concept is called a banker crop. So you essentially provide it nectar and flowers and other food sources, and then that parasitoid is built up in sufficient number, then it goes in. And the same thing with predators, right? So I've seen folks that are either releasing Japanese beetles or just not Japanese beetles, oh my goodness, releasing, not Japanese beetles, releasing, I'm blanking, all sorts of predatory beetles, but you can actually have them grow up on, an aphid population for a species that doesn't move off your crop, and then release them when needed, right? So that you can build that biological control up. I've also seen very successful, robust bat boxes. In California, they've done some analyses on whether birds were more important, were more of a pest or a benefit for something like strawberries. I don't think they'll have the same effect. Turns out it was about equal. So birds were providing as much damage as pest control as they were doing. So it was a total wash in strawberries, in organic strawberries, but that might not be the case in some of these systems. But absolutely, there are things you can do to support your kind of intrinsic biocontrol.
So would you have a list of crops that we could be growing as part of our system that would reduce the risk to the whole system?
I think diversity is the key piece. So the more types of crops, the less that moth is going to see that giant block of whatever it likes eating and recognize it as food and then come into that system.
But even like diversity of, I'm thinking of like the alyssum and I know it's harder in grain crops, but like alyssum with lettuce production acres, like spatial diversity or interseeding or buckwheat might have a different.
So if you interseed or if you have kind of companion crops that are grown together, those are great for insect mitigation. And they're fantastic because it means that the pest, which is used to seeing monocultures and rolling through, will not recognize your field as its ideal food source. So that mobile stage, that migratory stage, in this case, the adult moth would see that field and say, that cornfield over there looks much more tasty. There's more readily available food for it to eat. And that really increases resiliency in your system by having that diversity of crops.
When it comes to armyworms, would the same thing apply if you had a green crop growing between a corn row? Would that matter to an armyworm or not?
I think if you interseeded with a grain, probably less so because they also eat grain, but it would probably be less attractive than just having adult moths flying to corn. Yeah. I know I'm on a project where we actually interseeded with alfalfa, where we used alfalfa, kind of the first year alfalfa establishment happened in corn, and we actually saw some reduced leafhopper numbers. So I'm not sure on armyworms, we didn't look.